Hello, my dear students. Myself, Dr. Pitambar Humane, founder Botany for You, and working as Professor and Head Department of Botany, Dharampeth MP Dev Memorial Science College, Nagpur. First of all, I welcome you all in this online platform for the students of Life Sciences Botany for You. Friends, in today's lecture, we will conduct the different questions based on the B.SC Part 1 syllabus, that is Semester 1 syllabus of Botany Paper 1, prescribed by the Rtmnu Nagpur University, Nagpur. Though this lecture is based on the Rtmnu Nagpur University, Nagpur B.SC Part 1 syllabus, it will be also useful to the students of other universities where the similar topics are included. This lecture includes the different questions which are asked in last 5 years university examinations, and hence I am very much confident and hopeful that this lecture will be helpful to the students of B.SC Part 1 as well as the teachers teaching in UG classes. In this paper there are 4 different units which includes the microbes, that is viruses, prokaryotes, cyanobacteria and algae. And in the 4th unit, it contains the syllabus based on skill development, that is biofertilizer. So in the first unit, the viruses and the prokaryotes, that is the viruses, mycoplasma and bacteria are included. In the second unit, cyanobacteria and some general topics of the algae, that is economic importance, general characters of algae and classification is included, and the nostoc is included in the cyanobacteria. In third unit, the life cycle pattern of the different genera, that is Cara, Vaucaria, Ectocarpus and Batrachospermum is given. While the 4th unit, that is skill development, and that include the biofertilizers, in which the different topics, that is the scope and importance of the biofertilizers and the microbes used and commercial production of the microbes useful in production of biofertilizers. So this will be the complete syllabus of this paper. The pattern of questions which are asked in the university examination is fixed, that there are the 5 different questions: question number 1, question number 2, question number 3, question number 4 and question number 5. Each question carry the 10 marks and hence the total marks becomes 50. As you can see here, the question number 1 is based on the different topics of the unit 1, question number 2 on the unit 2, question number 3 on the unit 3 and question number 4 on the unit 4. These are the similar type of the questions. That first question contain the 2 notes, that is A and the B, which are 5 marks each, and in R to 8 there are the 4 different notes C, D, E and F, again from the same unit, but these are for 2.5 marks each. So in this way, either you can solve these 5 mark questions or you can solve the 4 different questions of 2.5 marks, and all these 4 questions are similar type or in a similar pattern and are based upon 4 different units. While the 5th question contain the 12 different notes and 3 from each unit, that is first 3 from the first unit, second 3 from the second unit, third 3 from the third unit and last 3 from the 4th unit. And you have to attend any TET out of these 12. You are suggested to solve all the 12 notes. and each one provided 1 mark. And in this way, again, the question number 5th will be 10 marks. And hence, each paper provided with the 50 marks. In first unit, that is viruses and prokaryotes, in which the first topic is virus. As we have seen earlier in the question paper, there are 3 different types of the questions: 5 mark question, 2.5 mark question, and 1 mark question. So first of all, in the viruses, we will see the 5 mark questions. These are the different questions which are already asked in earlier university examinations. Number 1, that is a general characteristics of the viruses. So we have to provide here the general characteristics of the viruses. That viruses are the obligate parasite. They can only reproduce or can use the energy when they are present inside the host. They show their living nature inside the host only, and when they are present outside the host, they are just in non-living particles. So in this way, we have to provide the general characteristics of the viruses in such type of the questions. The second question for the 5 marks is ultrastructure of the tobacco mosaic virus. Many times the students are doing the mistake in such type of the questions where they are only providing the theory and not drawing the diagram. But actually there are the marks to the diagram as well as the write-up. If you write correctly the complete data, and if there are the 3 marks on the write-up and 2 marks on the diagram, then you will not get these 2 marks because you have not drawn the diagram. And hence you are expected to draw the diagram as well as to provide the description of the cell. Hence, when such type of the question is asked, you have to draw this diagram of the tobacco mosaic virus. That it is rod shaped, having the RNA in the center, which is single stranded and coiled structure, around which there are the many protein subunits. And in this way, there is formation of cylindrical type of the structure. You have to provide complete detail of the structure of the TME in this question, including the diagram. In third question, that is the structure of T4 bacteriophage. This is a diagram which is expected when the question is asked on the T4 bacteriophage. Here there is a presence of the head and the tail, and tail having the collar, sheath and the base plate, and the base plates are provided with the tail fibers. And the DNA is present in this bacteriophage. Keep in mind, the tobacco mosaic virus, this one is a RNA virus, and the bacteriophage is a DNA virus. Then the fourth type of the question, that is the lytic cycle in T4 bacteriophage. In T4 bacteriophage, the type of reproduction, or we can say that the multiplication of the viruses is through the lytic cycle in case of the bacteriophages. So these are the complete stages of the bacteriophage which multiply itself when introduces its DNA in the bacterial cell. So first of all, there is attachment to the bacterial cell, or we can say that adsorption, then insertion of the DNA inside the host, then there is a replication of the DNA of the phage inside the host, that is bacterium, then synthesis of the different material like the proteins, head protein, tail fiber, and the protein which are used for the formation of the base plate, tail fibers, sheath, collar, etc. And when there is formation of such type of the different proteins, then there is formation of the assembly, and then there is release of the different T4 bacteriophages. So this is a complete lytic cycle. So here also in this question, you are expected to draw all these diagrams and to provide the different stages of the lytic cycle. In fifth type of the question, that is nature of viruses. Here in this question we have to provide the living as well as non-living nature of the viruses. If you see the characteristics of the living, then it must be made up of cell. It show the reproduction and it can absorb the energy or can take the food from its environment. These are the basic three different characteristics of the living organisms. If you see the viruses, they can show the multiplication or they are using the ATPs from the host organism. Though they are non-cellular, they show the reproduction as well as they are using the different enzymes from the host cell. So they show the living characteristics. But when they are present outside the host, then they do not carry out any activity and hence they are showing the characteristics of non-living things. So such type of the different characters which favor the living and non-living things characteristics of the viruses, that we have to provide in such type of the questions. The second type of the questions on viruses for 2.5 marks includes the living nature of viruses and the second one, that is nature of viruses, that we have already seen in a 5-mark question. So on this basis of this, you can see the same type of the questions are asked sometimes for the 5 marks as well as for the 2.5 marks. Then there is another type of the question, that is the plant viruses, again for the 2.5 marks. Here you can provide the tobacco mosaic virus. Then economic importance of viruses. In such type of the question, you have to provide the negative as well as the positive importance of the viruses. Say for example, the bacteriophages are used for the transfer of DNA from one bacterium cell to the another. Even the viruses are very much useful in genetic engineering. So these are the beneficial economic importance. But when we have to see some other organisms, they are responsible for the different diseases of the plants, animals and also human beings. So such type of the economic importance we have to provide in this question. Then question number 5, that is the structure of T4 bacteriophage, that we have seen in the last question also. Means the same question can ask for the 2.5 marks or the 5 marks. Even only this diagram is also asked for the 2.5 marks. Then 6th one, that is the ultrastructure of tobacco mosaic virus, diagram only. Means we have to draw only this diagram and you will get a full mark, that is 2.5 mark. Then in next one, that is the lytic cycle in bacteriophage. Again, the same question was asked for the 5 marks, and here you have to provide the similar account but in a short as it is asked for the 2.5 marks. Another question again same on the lytic cycle, but it is asked for 2 only diagram for the 2.5 marks. Then the 9th one, that is the animal viruses. So we have to provide the account regarding the animal viruses, just like to that of the 3rd question, that is the plant viruses. Then 10th one, that is the prophase. Prophase means it is the virus along with its capsid. Means it's a genetic material that may be either DNA or RNA, and its capsid or the covering which is made up of the protein. Then lysis. Lysis is nothing but the process in which the bacteriophages digest the bacterial cell to release its assembly in the form of T4 bacteriophages. So we have to provide this account in the lysis. Then if just virus is asked, you have to provide the account regarding the nature of viruses as well as the different type of the viruses. In third type of the question, that is 1 mark question on the viruses, which include the animal viruses, plant viruses and bacteriophages. Very commonly the plant viruses are RNA viruses, bacteriophages are the DNA viruses and animal viruses are the RNA as well as DNA viruses. No doubt there are again different types, but these are the very common things. So we have to provide the account regarding the animal viruses, plant viruses and the bacteriophages from the syllabus. Already the 1 mark question is provided in the form of 2 to 3 lines only, means that diagrams are not necessary. So in this question you have to write only 2 to 3 lines. Then lytic cycle, you have to provide the meaning of the lytic cycle in such type of the 1 mark question, that how after the formation of the assembly by the T4 bacteriophages inside the bacteria, they lyse or the digest the cell wall at the plasma membrane to release the assembly. Then TMV, that is the tobacco mosaic virus, which is the RNA virus and is responsible to cause the disease on the tobacco plant, that is the mosaic disease. Then obligate parasite, the viruses are the obligate parasite because to complete the life cycle there is need of the host organism, and without host it is not possible to complete the life cycle by the viruses, and hence they are known as the obligate parasite. Then capsid. Capsid is a protein covering which is present around the nucleic acid of any virus, and each capsid is made up of many capsomeres, and each capsomere is made up of number of protein subunits. Again, the capsid, that is the capsomere. Capsomere is nothing but a unit which is used for the formation of the capsid. In earlier question already we have seen that the prophase, that is the nucleic acid along with the protein which is present around it, that is known as the prophase. Then collar, that is a ring like structure which is present in a T4 bacteriophage, and that is known as the collar. Then adsorption, during the process of lytic cycle or the multiplication of the T4 bacteriophages, first of all the bacteriophages attaches itself on the cell wall of the bacterium, and this process of attachment is known as the adsorption. Then virion. Virion is a structure which contain the nucleic acid, either DNA or the RNA, and the protein which show its virulent nature, means able to infect on some other organism, and such type of the viruses are known as the virions. Again, the lysis, that is nothing but the process in which the T4 bacteriophages digest the bacterial cell to release its assembly outside the host cell. The second topic in the first unit, that is the mycoplasma. If you see the mycoplasma or the characteristics of the mycoplasma, mycoplasma is slightly larger than the viruses and smaller than the bacteria. In the evolution also, it is present in between the viruses and mycoplasma. If you see the difference in between the mycoplasma and the bacteria, then just it is without cell wall. So there are the different type of the questions on the mycoplasma, that is 5 mark question, 2.5 mark question and 1 mark question. So very commonly the question on the mycoplasma for the 5 marks which is asked in earlier examinations by the university, that is structure and reproduction in mycoplasma. Then second type of the question asked is the morphology and properties of the mycoplasma. So here in a structure or the morphology, or even the third type of the question, that is the mycoplasma, you are expected to draw this diagram or the figure. that here the three-layered plasma membrane, which is lipoproteinous in nature, which contain its genetic material, that is the DNA, then ribosome containing the soluble RNA and some soluble proteins. So you have to provide this diagram in all these three types of the question. Along with, you have to provide the account regarding the reproduction, and reproduction of the mycoplasma is by means of binary fission, and the second one is by means of budding. So these two common methods of the reproduction in the mycoplasma are found. Then in 2.5 marks questions, same type of the questions are asked, that is structure of the mycoplasma, then ultrastructure of mycoplasma. Again, both the questions are asked in a different manner, but the answer is same. We have to draw the diagram of the mycoplasma and provide the account or the structural details. Then properties of the mycoplasma. Here we have to provide the different characteristics of the mycoplasma. Then reproduction in a mycoplasma, again the same question that is asked for the 5 marks. We have to provide that the reproduction in the mycoplasma is carried out by means of the binary fission as well as the budding. And the most common question which is asked on the mycoplasma for the 1 mark in different question papers by the university, and that is the PPLO, Pleuropneumonia-like organisms. The third topic in this unit, that is the bacteria. In bacteria, there is a question that is the ultrastructure of bacterial cell. So here in a figure you can draw this figure which show the flagella, then presence of the cell wall, cell membrane, then presence of the plasmid, chromosomes, ribosomes, then cytoplasm, pili and the flagella. So in this way we have to provide the complete details of the bacterial cell and also to draw the diagram. Then reproduction in bacteria. Though there are the different type of the reproduction in bacteria, but we have to provide the 2 different types as they are provided in the syllabus. Number 1 is binary fission and second one the conjugation. So we have to provide these 2 methods of reproduction in case of the bacteria. Then third question for the 5 marks asked in the bacteria, and that is economic importance of bacteria. As we have seen earlier in case of the viruses, here also we have to provide the negative as well as the positive importance of the bacteria. The very common use of the bacteria in making the curd, as well as when we have to multiply the gene, when it is transferred in a plasmid, then again the bacterial cells are used. So bacteria again are responsible to cause the different plant diseases, animal diseases and the diseases on the human beings also. Means here also there are the positive as well as the negative importance. So we have to provide all this one. In 2.5 mark questions, again there is a question on the binary fission in the bacteria. So these are the series of diagrams which show the binary fission. So here you can see, here there is a replication of the genetic material or the DNA first by means of the amitosis. Then there is formation of a cell plate in between the genetic material, and hence resulting into the formation of the 2 cells from the single cell of the bacteria, and hence there is multiplication of the bacterial cell and hence called as the reproduction. In second one, that is conjugation in bacteria, again this question is asked for the 2.5 marks. So this is a conjugation. Conjugation is considered as a sexual reproduction in bacteria, in which there is a mating in between the 2 bacterial cells by means of the sex pili. one cell considered as a male and another cell considered as a female, that is F positive and F negative. After conjugation, both the cells becomes F positive. So we have to provide these all the diagrams and the complete matter regarding the conjugation. Third question, that is the ultrastructure of bacterial cell and diagram only, means here this diagram we have to provide in this type of the question. Then fourth one, that is the flagella in bacteria, that is monotrichous, amphitrichous, lophotrichous, etc. on the basis of the development of the type of the flagella from the cell wall. Then fifth one, that is the economic importance of the bacteria. The same question was asked for the 5 mark and the same question is asked here again for the 2.5 marks. So depending on the marks, we have to provide the account for the questions. Then useful aspects of the bacteria. Earlier we have seen that in economic importance we have to provide the negative as well as the positive importance, but here specifically the useful aspects are asked in the question and hence you have to provide only the useful aspect, that it is useful in making the curd, it is used in the genetic engineering, it is used to transfer the gene from one cell to the another cell. So such type of the use we can provide in this type of the questions. In 1 mark question, there is sex pili. Already we have seen that in a conjugation there is use of the sex pili for the formation of the conjugation tube through which the DNA molecule of the plasmid transfer from one bacterial cell, that is F positive, to the another bacterial cell F negative. Then binary fission is the process of the reproduction in case of the bacteria, in which there is formation of the 2 cells from each cell. Then flagella is a locomotory organ in case of the bacteria. There may be presence of the 1 flagella, there may be presence of the 2 flagella or the many flagella. Then mesosome, that is the in certain places in a bacterial cell there is infolding of the plasma membrane which is provided with the many enzymes, and hence these enzymes are involved in the different processes and also it play an important role during the multiplication of the cell like the binary fission. Then prokaryotes is the general words in which we can say that when the nucleus is not provided with a nuclear membrane, then such type of the organisms are known as the prokaryotes. So bacteria and cyanobacteria, these are the members which are included in prokaryotes. In second unit, that is based on the cyanobacteria and the algae, and first one is a cyanobacteria. On cyanobacteria there are the again 5 mark question, then 2.5 mark question and 1 mark questions. The 5 mark question include the reproduction in nostoc and ultrastructure of cyanobacteria. If you see the any question paper in last 5 years, then you will always find the 5 mark question that is on the reproduction as well as the ultrastructure of the cyanobacterial cell. So when the question is asked on the ultrastructure of cyanobacteria, then you have to draw this diagram and to provide completed details regarding the cyanobacterial cell. If the question is asked on the reproduction, first of all you have to remember in a nostoc or the cyanobacteria the sexual reproduction is absent, and hence here the asexual or the vegetative type of the reproduction is there, which includes the reproduction by means of the fragmentation, second one by means of the akynates, third one by means of the heterocyst, and fourth one by means of the hormogonia. So just you have to remember that there are the 4 different types of the reproductions in a nostoc or the cyanobacteria. In the 2.5 mark questions, there is again same question, that is the ultrastructure of the cyanobacterial cell, but here only the diagram is asked. So you have to just draw this diagram in this question. Then economic importance of the cyanobacteria. We have to provide the different negative as well as the positive economic importance of the cyanobacteria in this question. Basically the cyanobacteria are responsible for atmospheric nitrogen fixation. So this is a most important function of the cyanobacteria, which is carried out because of the presence of the heterocyst in the cells of the nostoc or the cyanobacteria. Then thallus structure of the nostoc for the 2.5 mark question. So here this is a diagram. You can draw the three different type of the cells, that is the vegetative cell, achaenates as well as the heterocyst, and each cell is bounded by the mucilaginous sheath. Again, the heterocysts are the empty looking type of the cell and they are devoid of the chloroplast. So this diagram along with its description is expected in a thallus structure of the nostoc. Fourth question is asked, that is the structure of heterocyst. In this question, you are expected to draw this diagram of the heterocyst, that here there are the presence of thylakoids, then outer layer, presence of the plasma membrane, then here there are the presence of the polar nodules on both the sides. So this structure or the diagram along with the description is expected in structure of heterocyst. Then general characteristics of the cyanobacteria, you have to provide the matter regarding the characteristic features of the cyanobacteria in this question. Then in a 1 mark question, that is the polar nodule, you know, when there is a attachment of the heterocyst to the adjacent cell and there is a channel, then this channel is provided by the heterocyst, and this channel is present on the polar nodule. Means the polar nodule is only present on both the sides when the heterocyst is intercalary, and when the heterocyst is terminal, the polar nodule is present only on the side where the vegetative cell is attached to it. Then achaenates are the resting spores when there is unfavorable initiation of the unfavorable conditions, that is increase in the temperature, evaporation of the water from the water bodies, there is formation of achaenates by storing the food material in some of the cells. And these achaenates, because of the presence of stored food material, on reaching or on next season, there is formation of the new organism, that is it involved in the process of reproduction. Then Hormogonia is a structure or the filament of the nostoc or the segment of the nostoc filament, in which there are the presence of two heterocysts at both the sides, and rest of the vegetative cells are present in it, which are provided with mucilaginous sheath. Again, the Hormogonia are involved in the process of reproduction. Then heterocysts are the special type of the cells. Already we have seen that the empty looking cells which are involved in the process of nitrogen fixation. Then mucilaginous sheath, the colony is encircled by the mucilaginous sheath, as well as each cell or the individual filament of the nostoc is encircled by the mucilaginous sheath. So such type of the different questions for the 1 mark from the cyanobacteria or the nostoc are asked in the university examinations. The second topic, that is the algae, and on algae there are the typical type of the 5 mark questions, that is general characteristics of the algae. Several times this question is asked in the university examination. Then economic importance of the algae, this is also asked several times in the university examinations. And sometimes the classification of algae provided by the Frisch in 1954, that is also asked for the 5 mark. So these are the 3 different type of the questions from the algae are asked for the 5 marks. For 2.5 marks, basically the general characteristics of algae, see the same which was asked for the 5 mark. Then economic importance, again the same question asked for the 5 marks. Means both these questions are asked for the 5 marks as well as the 2.5 marks. For 1 mark, there is question on the algal blooms or the water blooms. So in certain cases on the water body, there is a growth of algae on the surface of the water, or we can say that the excess growth of the algae. Such type of the excess growth of the algae on the water body is known as the algal blooms or the water bloom. This is because of the presence of a specific type of the nutrients in the water. Second one, that is the pyrenoid. Pyrenoid is again one of the characteristic features of algae. In algal cells, there is a proteinous bodies and that is known as the pyrenoids. In 2.5 mark questions or the 1 mark question that are asked on the different classes which are provided by the Fritsch in his classification. In Fritsch classification, there are 11 classes from the chlorophyceae to myxophyceae. That is here the chlorophyceae, xanthophyceae, chrysophyceae, bacillariophyceae, then cryptophyceae, dinophyceae, chloromonadinae, then euglenophyceae, then phaeophyceae, rhodophyceae and myxophyceae. So any one of these can ask for the 2.5 marks or 1 marks, and if the question will be asked on the classification of algae for 5 marks, then we have to provide the characteristic feature of these different classes. Here again the 1 mark question will be on grass green algae, yellow green algae, diatoms, yellow or the golden brown algae, dark yellow algae, then brown algae, again bright green algae, then pure green algae, brown algae, red algae or blue green algae. So even such type of the words can also be used to ask the 1 mark question in the examinations. In third unit, as we have seen, there are the different genera from the algae, in which the first one, that is the Chara. The Chara is the most advanced algae and it is found in the fresh water or in a shallow water bodies like the lakes. In 5 mark questions, it is asked that is the sex organs in Chara. Specifically, if you see the Chara, then the sex organs are developed from the nodal region of the branches of limited growth. So here if you see here the branches, then you will find that on the branches of limited growth from the nodal region there is a development of a globular structure or spherical structure which is known as the antheridium or the globule, and on the same node there is a development of a female sex organ, that is nucleus or the oogonium. So we can either provide both in case of the sex organs in Chara, that is the structure of globule and structure of nucleus. If the question is asked only on the globule, we have to draw the diagram of the globule only, and if the question is asked on the nucleus, we have to draw the diagram of the nucleus only. In all these 3 questions, the diagrams from the globule and nucleus are most important and are expected. In 4th type of the question, that is the nucleus and the globule in Chara, the same question, that is the sex organs in Chara, is the same. Here only the sex organs names are given. Again the same question as asked in the examination, that is the LS, that is globule and nucleus in the Chara. So again this type of the questions for the 5 mark from the Chara asked earlier in the university examinations. For the 2.5 marks, again there is a question on the globule in Chara, that is a diagram only, then nucleus in Chara, then thallus of Chara. See this is somewhat different, means we have to provide the thallus structure of the organism, that is presence of the rhizoids from the lower nodes, presence of the 2 type of the branches, that is the branches of limited growth, branches of the unlimited growth, and the development of the scales or the stipulates from the nodes. So complete these details are expected to provide in this question. In 4th one, that is the vegetative reproduction in Chara. No doubt here the sexual reproduction in Chara is advanced oogamous, that is development of the male sex organ, and female sex organ, that is the globule and the nucle that we have seen. Again, there are the different methods of the vegetative reproduction in the Chara that we have to provide. That is by means of the development of the bulbils, which are either developed from the stem or the root. Again, there is a development of a bunch of small bulbils that are amorphous bulbil, and certain special type of the structures are developed, that is the star-shaped bodies containing the stored food material, that is the starch, and these are known as the amylum starch. So these are the different methods of the vegetative reproduction. Then post-fertilization changes in Chara. After the sexual reproduction, that is the fusion of the male and the female gamete, inside the nucle, here it divide by means of the meiosis into 4. Out of that, here 3 degenerate and only 1 remain functional, and this is only involved in the formation of the new plant body, and hence usually this type of the question is asked in case of the Chara. In one more question in the Chara, the amylum stars, that here this one. Again, for the 1 mark question, you are not expected to draw the diagram. Only we have to provide the answer in 2 to 3 lines only. Then amylum stars, that is the starch which is named amylum, and they are star shaped and hence known as the amylum stars. They are developing from the nodal regions of the stem, that is the lower nodes, as well as the roots or rhizoids. Then secondary protonema, that is the protonema like structures which are developing from the nodal regions of the stem or the axis, and they are known as the secondary protonema. Again, it is a method of vegetative reproduction in case of the Chara. Then bulbils are also developed from the axis as well as the rhizoids from the nodal regions, and after detachment from the plant body, they regenerate into the complete organism or the thallus. Then small bodies, that is the amorphous bulbils. You can see here the bulbil, stem bulbil, root bulbil, and amorphous bulbil. So amorphous bulbil are the small tuberous bodies which are developing from the nodal regions of the axis or the stem. Then globule, which is the male reproductive organ developing from the nodal region of the branches of limited growth, and it is just present below the nucle. Then again, the nucle is a female reproductive organ developing from the nodal region of the branches of the limited growth, and it is always grow above the globule. Then corona, these are the special type of the cell which are present around the nucle, and they are free at the end of the nucle, and that is known as the corona. Then homothallic, here homo means the same and hetero means the different. Means when the male reproductive organ and the female reproductive organ are present on the same plant, then it is known as the homothallic condition. Here, if you see this diagram of the Chara, here the sex organ, that is the antheridium and oogonium, both are developing from the nodal region of the same plant, and hence such type of the species in which the male and the female reproductive organs are developed on the same plant, that is known as the homothallic. Then monoecious. In a monoecious, the same type of the meaning is there, that when the sex organs, that is the male and the female sex organs, are developing from the same, developed on same plant, then it is known as the monoecious condition. Here in homothallic or the monoecious, here the example is Chara. Then oogamy. In oogamy, there is a development of the oogonia, which is the characteristic feature of the Chara, and in oogamy, the female sex organs remain at its place and the male gametes move towards it. Then stipuloids, here these are the scale-like structure which are present in the nodal region of the Chara. Then stoneworts, as if you see these algae, the certain chalky compounds that is the calcium containing material, is deposited on the cell wall, and hence it is just a hard plant, and hence it is known as the stone buds. Then capitulum, if we see the structure of the antheridium or the globule, then there is presence of primary capitulum, secondary capitulum, and from the secondary capitulum there is development of the antheridial filaments, and hence capitulum is a structure which is present inside the globule in case of Chara. The second genus, that is Vaucaria, and if we see the Vaucaria, which is different than that of the Chara, as the Vaucaria is a single cell algae, means it is unicellular algae. Though it has the many nuclei, but the thallus is cenocytic, that there is no partition or the septum in between the nuclei. In 5-mark question on a Vaucaria, there are the most common question, that is the asexual reproduction in Vaucaria, and second one, that is the sexual reproduction in Vaucaria. In case of asexual reproduction, we have to provide the reproduction by means of formation of a planospores, and second one, that is the zoospore. These are the two common type of the spores formed by the Vaucaria in case of asexual reproduction. In second question, that is the sexual reproduction in Vaucaria. Here you can see the male reproductive organ, that is the antheridium, and the female reproductive organ, that is the oogonia. They are developing from the same thallus side by side in case of the homothallic species or the monoecious species. So we have to provide the structure of antheridium, structure of oogonium, and the reproduction that is expected in the sexual reproduction in Vaucaria. In 2.5-mark questions, that is the thallus structure of Vaucaria, here we have to draw this diagram and provide the complete description of the thallus, including the portion of filament showing the internal structure. Then sex organs of Vaucaria, the same question, that is sexual reproduction in Vaucaria, we have to provide the details regarding the structure of sex organs in Vaucaria. The third question asked in case of the Vaucaria, that is a Gongrosira stage. If you see this diagram, you will find that this cenocytic structure of the thallus is converted now in a multicellular type of the condition. Earlier, it was considered that there is a Gongrosira algae, and this stage of the Vaucaria just look like to that of the Gongrosira algae. But later on, it is found that there is no such type of the algae, and it was just a one-off stage of the Vaucaria. But from then, here this type of the structure in which the protoplasm of the Vaucaria thallus is converted into number of segments, or we can say that it is a akinets. So when such a thallus is converted forming the akinets or the cyst or the hypnospores, then this structure is known as the Gongrosira stage of the Vaucaria. In 1-mark question, which is asked for the zoospores and planospores, there is no any difference in between the formation in between these two type of the spore, that is zoospores and the planospore, except there are the presence of flagella in case of the zoospore, and here there is no flagella in case of a planospore. Just you have to remember this difference. Again, the type of the formation of the spore is same. Then cenocytic thallus. Earlier we have seen that there are the many nuclei, but here there is no partition wall, in between the different nuclei which are present in the thallus of the Vaucaria, and such type of the structure is known as the cenocytic thallus. Then fourth one, Gongrosira stage, as we have seen earlier in the 2.5 mark, that when the protoplasm of the Vaucaria thallus is divided into number of akynates or the chain of the akynates, then it is known as the Gongrosira stage, as it look like to that of the Gongrosira algae, but it is not actually the different algae, it is the same stage of the Vaucaria. Then fifth one, that is synzoospores. Here the zoospores of the Vaucaria are provided with many flagella, and two flagellas are developing from each nuclei, and hence it is considered that it is a combination of the many zoospores forming a single structure, and hence it is known as the synzoospore, which germinates and give rise to the new thallus of the Vaucaria. Then wonderplasm. If you see the wonderplasm, during the course of the development of the female reproductive organ, there is formation of such a type of the protuberance on the thallus of Vaucaria, and some of the nuclei and the cytoplasmic material is accumulated in this protuberance, and this is known as wonderplasm, and it is very commonly asked for the 1 mark in case of the Vaucaria. Then seventh one, that is the akynates or the hypnospores or the cyst. As we have seen a Gongrosira stage, here these are the different akynates or the hypnospores, or it is also known as the cyst, as these are the resting spores which are commonly formed at the end of the favorable season or on onset of the unfavorable season. The third algae in this unit, and that is Ectocarpus. In Ectocarpus, again the same type of the questions are asked for the 5 marks, that is the asexual reproduction in Ectocarpus, and second one, that is the sexual reproduction in Ectocarpus. Means both type of the reproductions are asked for the 5 marks. In asexual type of the reproduction, here it is by means of the formation of zoospores, and it is on diploid plant. Means here in Ectocarpus there are two different plants, morphologically similar but they are genetically different. That there are the sporophytic plants, that is the diploid plant, and the gametophytic plant, that is a haploid plant. So asexual reproduction is carried out on a diploid plant, and sexual reproduction carried out on a haploid plant or the gametophytic plant. So we have to provide the development of the zoospore in case of the asexual reproduction, and development of the gametangia in case of the sexual reproduction, in which there is a formation of the gametes or the isogametes, and then there is a fusion in between the gametes of the plus strain and the minus strain. So here we have to provide the complete description regarding the sexual reproduction. In case of the 2.5 mark questions, there is a question on the unilocular sporangia or the plurilocular sporangia. During the formation of zoospore, there is a formation of plurilocular sporangia on the diploid plant body, and these zoospores are formed without meiosis, and hence all the zoospores are diploid, and hence again they germinate to form the same sporophytic plant body. But when there is a formation of the unilocular sporangia, there is a formation of the zoospore which are myozoospores, and they are formed after the reduction division, and hence they are haploid. Means after germination there is formation of the new plant and that is a gametophyte. Means in case of the plurilocular sporangia, it results into the multiplication of a diploid plant, and in case of the unilocular sporangia, it germinates to form the gametangia, that is the haploid plant. Then third one, that is the thallus structure of the ectocarpus. Here the characteristic feature of the ectocarpus is that the type of the cell in an ectocarpus, they are barrel shaped with a discoid type of the chloroplast. Presence of rhizoids is again one of the characteristic features of the ectocarpus. Then in a 1 mark question, here there is a clump formation. If you see the sexual reproduction in ectocarpus, you will find that there is a one gamete which is a female gamete which is encircled by a many male gametes, and this structure is known as a clump formation. Then second one, that is the unilocular sporangia. Earlier we have seen that the unilocular sporangia are developed on the sporophyte, but during the course of the development of the meiospore there is a meiosis, that is the reduction division, which result into the formation of the meiospore, which germinate to form the gametophyte or the haploid plant body. Then plurilocular sporangia is responsible for the formation of the zoospore, which are diploid in nature and are involved in the multiplication of the sporophytic plant body. Again, the meiospore, that is inside the unilocular sporangia, there is a formation of the meiospores. And on the gametophytic plant body, there is a development of the gametangia, which is structurally similar to that of the plurilocular sporangia. But instead of the formation of the diploid zoospore, which are formed in the plurilocular sporangia, there is formation of the zoospore like structures which are the gametes in a gametangia, which are haploid in nature. The fourth one algae in this unit, that is a batrachospermum, which is a beautiful in its appearance and found in flowing water in a shallow water bodies. In a 5 mark question, there is a question on the sexual reproduction in batrachospermum, and the second type of the question, that is the sex organ in batrachospermum. Though the questions are asked in a different pattern, but almost similar type of the description is expected in both type of the question. In second one, that is the sex organ in batrachospermum, we have to provide the structure of the male reproductive organ and the female reproductive organ. But in case of the sexual reproduction, we have to provide the formation of the gametes, fusion of the gametes, and again there is a formation of the spores, that is the corpospores. Then in case of the 2.5 marks, there is a question on the thallus structure in a batrachospermum. Just like to that of the Chiara, here also the type of the branches in batrachospermum are branches of the limited growth and the branches of the unlimited growth. In Chiara, there is a development of the branches of limited growth in one direction, but here in case of batrachospermum, the branches of limited growth develops on all the sides, and because of that there is a formation of a globular structure around each node, and that is known as the glomerule, which is frequently asked for 1 mark in the examination. In case of the sex organs in batrachospermum, as we have seen in the 5 mark question, it is also asked for the 2.5 mark question. So here we have to provide the male reproductive organ, that is the spermatangium, and the female reproductive organ, that is the corpogonium. So we have to provide the structure of both of these one along with its description. Then asexual reproduction in batrachospermum. After germination of the carpospore, there is development of the juvenile stage in case of the batrachospermum, which reproduces by means of the monospores, and reproduction by means of the monospore is a method of asexual reproduction in batrachospermum. The 1 mark question in batrachospermum include the glomerule, as we have seen that when there is a development of the branches of limited growth in case of the batrachospermum, from all the side. Here there is a formation of a spherical type of the structure from each node, and this is known as a glomerule. Then frog spawn. If you see the complete thallus of the Batrachospermum, we will see here it is a beaded structure, that is, the beads are on the string. So as they are present inside the water, a jelly-like structure is observed inside the water when there is a Batrachospermum, and it is just similar to that of the eggs of the frog, and hence it is known as the frog spawn. Then third one, that is the Carpogonia. If you see the Carpogonia, here there is a, after the fertilization, the third one, that is Carpogonia. The Carpogonia is a female reproductive organ of the Batrachospermum, which is usually a flask-shaped structure in which there is a presence of a single nucleus at the base of the Carpogonium, which act as a egg. Then fourth one, that is the trichogyne. The terminal part of the Carpogonium is known as the trichogyne, which is almost hair-like structures and colorless, and is used to receive the male gametes or the spermatia. Then the spermatia or spermatogonia, these are the unicellular structures which are formed by the Batrachospermum as a male gametes. Then corposporophyte. Corposporophyte is the structure which is formed after the sexual reproduction, that is after the formation of the zygote in case of the Batrachospermum, when there is a fusion of male and the female gamete and formation of zygote. Zygote divide and redivide, and later on it result into the formation of the gonymoblast filament. And in this way, here from the gonymoblast filaments, there is a formation of a corpospores from its terminal side. So there is formation of a corposporangia from the gonymoblast filament, and the terminal cell is involved in the formation of corpospore, and hence it is known as the corposporangia. This complete structure of the fruiting body in case of the Batrachospermum is known as the cystocarp. As we have seen that the juvenile stage of the Batrachospermum reproduces by means of the monospore, and that is known as the Contrancia stage. So this is again the in Contrancia stage it reproduces by means of the monospore, that is by means of asexual reproduction. Unit 4 is based on biofertilizers, and this is a different unit, that is from the skill based syllabus. On this unit there are again the 5-mark question which includes the microbes used as biofertilizers. When this question is asked, then specially I have given here that you have to provide regarding the rhizobium and azotobacter, and both of these are the nitrogen fixing bacteria. That is, the rhizobium is found in association with the root nodules of the other plants, as well as the azotobacter, azospirillum and acetobacter are the free living organisms. Then azolla is a pteridophyte in which there is a presence of a cavity inside the leaf in which there is a presence of Anabina azoli. which is responsible for the atmospheric nitrogen fixation, and hence it is commonly used in the paddy field. Then cyanobacteria that we have seen in the unit 1, that is the nostoc, that is again used as a nitrogen source as a biofertilizer. Then mycorrhiza, and mycorrhiza is again used as a biofertilizer as it is involved in absorption of the different minerals. Then second question for the 5 mark on the biofertilizer is the commercial production of the biofertilizer. This question may be either on rhizobium or azotobacter or phosphate solubilizing bacteria or azolla. As we have seen that rhizobium form the root nodules on the leguminous plants. Azotobacter is a free living nitrogen fixing bacteria. Then phosphate solubilizing bacteria are involved in formation of the phosphates. Azolla is for the nitrogen fixation. In case of the 2.5 marks, here there are the importance of biofertilizer. Means we have to provide the use of the biofertilizer. That is, it is environment friendly. It reduces the pollution, as the chemical fertilizers are responsible for the soil pollution as well as the water pollution. Then scope of the biofertilizers, where we can use, how we can use. So here both the questions are again can combine to form the 5 mark question, that is the importance of biofertilizers and scope of the biofertilizers. Then phosphate solubilizing bacteria. As we know, the phosphate is made available by such a type of the bacteria or the microorganism. Even phosphate solubilizing microorganism or phosphate solubilizing bacteria is given the name. Then fourth type of the question for the 2.5 mark is a rhizobium as a biofertilizer. As we know that the rhizobium form a root nodules on the leguminous plant and provide the extra nitrogen to them. Then PSB, that is the phosphate solubilizing bacteria, as a biofertilizer. We have to provide the details regarding the PSB as a use of biofertilizer. Then azolla as a biofertilizer. Here this is a diagram of the azolla plant, and in which the cavity of leaf cavity is there, in which there is a presence of the Anabaena azulei, which is a cyanobacteria and is involved in the atmospheric nitrogen fixation, and hence very commonly the azolla is used as a biofertilizer in case of the paddy plant, where there is a presence of the ample of water. Then azotobacter as a biofertilizer. As we have seen, the azotobacter is a free living nitrogen fixing bacteria, and hence it increases the nitrogen content of the soil, and hence there is no necessity to provide the extra nitrogenous containing fertilizer or the chemical fertilizer to the plants if we provide the azotobacter as a biofertilizer. Then in case of the 1 mark question, that is asked for the biofertilizers. Biofertilizers are the organisms or the microorganisms or the bacteria which are provided to the plant as a nitrogen source. Then the phosphate containing or the to provide the phosphate, there is use of the phosphate solubilizing bacteria or the phosphate solubilizing microorganisms. Then Cryma test, when the different nodules are crushed, then they are grown on a special type of the medium that is known as the and is used as a Cryma test to know the development of the rhizobium and to separate them from the other organisms. Then third one, that is again the glucose peptone agar test. This is also used to know the growth of the rhizobium. Then azotobacter, that we have seen that it is a free living microorganism and is involved in the atmospheric nitrogen fixation. Next one is that is the rhizobium. Rhizobium is a cylindrical or the bacillus type of the bacteria and is involved in the formation of the root nodules of the leguminous plant and is involved in the atmospheric nitrogen fixation because of the presence of a special type of the structure. Then Azula, again a pteridophyte which is found in association with the cyanobacteria. Then top dressing, in case of certain plants, the microorganisms or nitrogen containing microorganisms or the free living microorganisms are mixed with a decomposed material or the decomposed manures and is provided directly to the plants which are grown close to each other, and such a type of the application of the microorganism is known as the top dressing. Then certain microorganisms are provided directly to the leaf and hence it is known as the foliar application. And very commonly the foliar application of the microorganism is provided to the mulberry plants. Then next one is the Yamma, that is the yeast extract mannitol agar. Here the microorganisms like the rhizobium is grown on the Yamma and it can be identified that it is rhizobium or the agrobacterium. Then next one is the curing. Curing is the process in which at a room temperature the microorganisms are multiplied. Then inoculation. Inoculation is the method in which the microorganisms are transferred on a culture media for the growth or the multiplication, and a method in which microorganisms are grown in a large scale, that is known as the mass culture. So in this way the 1 mark questions on the biofertilizers will be asked in the university examinations. You can prepare the 2 to 3 lines notes with you and can read or revise during the examination for such type of the 1 mark questions. As already I have provided that there is no necessity to draw the diagrams in 1 mark questions. If this lecture is useful to you, then kindly like, share and subscribe our YouTube channel and also press bell icon for the alerts of every lecture to be uploaded on Life Sciences titles. If you want to associate with us through the Telegram group, then the link is given in the description of this lecture. Also, you can visit our website www.botanyforyou.com, where also you can get the Telegram link. Thanks a lot once again for watching this lecture completely.